Hi folks, and welcome to another edition of Zog Science. Today we're going to be talking about enzymes and how they actually go about catalyzing chemical reactions. So recall that enzymes are catalysts. You learned that in freshman biology, and you probably learned about catalysts in chemistry as well in terms of some inorganic catalysts. Well, today we're going to be talking about enzymes, and they are biological catalysts. So they speed up chemical reactions, and an example of that would be sucrase. And sucrase is is an enzyme that speeds up the reaction of taking sucrose and breaking it down into its constituent parts, glucose and fructose. You'll notice that sucrase is an enzyme. ACEs, the ASE ending, that is generally an indicator that something is an enzyme. And you'll notice that that is also very similar to sucrose, right? So usually what the enzyme is named as is what it is actually going to be breaking down. Now, enzymes work by reducing the activation energy of a chemical reaction. Well, what is the activation energy? The activation energy is the energy that it takes to initially break the bonds of a reactant and to get it to a transition state. Transition states tend to be very unstable, and so therefore you have to put energy in order to make that happen. Now, we've talked about the fact that reactions occur spontaneously or non-spontaneously, depending on how the energy of the products compares to the energy of the reactants. Well, the transition state is always going to have a higher energy, and that energy usually can come from the surrounding environments, if it can, but that takes a lot of energy, right? So an enzyme is going to come in and it's going to reduce that activation energy in order for us to be able to get to our products. So again, what they're going to be doing is that they're going to be affecting the chemical reaction by reducing the activation energy. That doesn't change the amount of free energy of the products of the reactants. It just speeds them up and makes them occur quicker than what they would have normally, because it takes less of that activation energy. So here we've got our diagram. We can see the black line is the reaction without the enzyme. The red line is the reaction with the enzyme. Again, we see that the free energy of the reactants and the products is the same, so our delta G is the same, but the activation energy is lower. So what that means is that it takes less of that energy. Again, activation energy typically comes from the kinetic or the temperature, the kinetic energy or the temperature that is in the molecules themselves or in the surroundings. And so by lowering the activation energy, you're much more likely to have a reaction take place. It can also take place at lower temperatures. Now, enzymes are only able to function with specific substrates. The substrate is the reactant. It's the molecule or molecules that the enzyme is going to bind to. When the substrate and the enzyme bind, they create the enzyme substrate complex, and the active site is the region where the substrate binds with the enzyme. Enzyme specificity results because shape determines function, right? Or form determines function. That's one of our things that themes that we've talked about for this course. And the shape of an enzyme's active site is going to fit the substrate, and it's not going to be able to fit other things unless they're very, very similar. And that can lead to some issues. Certain drugs take advantage of this, things like that. Toxins also take advantage of this. But enzymes change their shape slightly, so it's not exactly that they fit perfectly, but they do kind of change their shape in order to fit around the substrate. We tend to call this the induced fit, or sometimes the glove model. When you put on a glove, the glove will stretch a little bit in order to fit your hand. And so that sometimes is, again, called the induced fit model. So the way that this will happen is that you'll have your substrates. Substrates will bind to the active site, forming the enzyme substrate complex. Then you'll have the substrates be converted into the products. The chemical reaction will take place. The products will then be released, and then you will have the enzyme. Enzymes can be reused. They don't get destroyed. They aren't changed by the chemical reaction that takes place, and so they can be constantly recycled and reused over and over again. Now, there are lots of conditions that affect enzyme activity. For example, pH and temperature, but also some things such as salinity can affect it, or sometimes even what the enzyme is in. And the reason why is that enzymes are proteins, which we'll talk a little bit about later on. And the reason why it matters is that proteins are made out of amino acids, and amino acids. Now, lots of things can affect enzyme activity. Those include temperature and pH. The reason why temperature and pH are going to affect that is that enzymes are made of proteins. And as we've learned, proteins are made up of amino acids, and amino acids have the side chains, and how the side chains interact is going to determine how the protein is going to fold. Well, when you change the temperature, it can cause some of the weaker side chain interactions to come apart. Or if you change the pH, that is going to either add protons or remove protons from those acidic and basic side chains, which is going to then change how they are going to interact with one another. And so we get what we call denaturing, where the enzymes will change shape. Now, coenzymes or cofactors are non-protein enzyme helpers. Basically, they help the enzyme to do their job. A lot of vitamins that you take are coenzymes or are the raw materials that coenzymes are made from, which is the reason why you've got to make sure that you are getting all of your vitamins. Now, there's two different types of inhibitors that can come into play as well. Inhibitors are going to bind to the active sites of an enzyme, which means that they're competing with the substrate. They basically make it so the substrate can't bind. Or we can have non-competitive inhibitors. And non-competitive inhibitors will bind to another portion of the enzyme. And when they do, they cause the enzyme to change shape. And when the enzyme changes shape, now it can't actually bind to the substrate. Lots of inhibitors are things like toxins or poisons, pesticides and antibiotics. And so all these kinds of things that have negative effects on living things, they are a lot of times impacting some sort of enzyme and preventing it from functioning, and therefore stopping a metabolic pathway that's critical for the living cell. Hi folks, and welcome to our next edition of Zog Science. Welcome to our second unit. So here's an example of inhibitors at play. A competitive inhibitor, again, is going to bind at the active site and prevent that substrate from binding. And then we have the non-competitive inhibitor that is binding somewhere else on the enzyme. It's causing that shape change at the active site, and therefore the substrate cannot bind. Enzymes can be regulated, and that can help to control metabolism, because remember, metabolism is all about metabolic pathways, and enzymes are necessary for those metabolic pathways to work. So whether or not you have the correct enzymes is going to affect whether or not you get the products. Feedback inhibition is where the end product of a metabolic pathway shuts down the pathway. So basically, when you produce something at the end, it is going to stop what's at the beginning. This is all sometimes called negative feedback. So basically, it prevents a cell from wasting resources, and it prevents from making too much of a product than is needed. Supply and demand. So here's an example of feedback inhibition. This is a enzyme which is going to take threonine and turn it into isoleucine. These are both amino acids. And so the end product, isoleucine, is actually a non-competitive inhibitor. And so it's going to bind to the enzyme that is going to start this process, and it's going to change the site, the active site, so that way the threonine cannot bind. And so basically, when you have high levels of isoleucine, you don't need more of it, so you stop the process of making more of it, which is good and efficient, and again, it prevents the cells from wasting resources. There are also structures within the cell that help to bring the order to this. Enzymes act as structural components of membranes, and so you want things to be being produced when you need them, but you don't need it necessarily for other things. You also want to make sure your enzymes are contained within the proper location. So for example, mitochondria, right? They have specific enzymes. You don't want those enzymes going somewhere else because they might interfere with other pathways. So enzymes are typically self-contained inside of organelles in order to help them perform their function, but not interfere with other functions. That's all I've got for you today. Thanks so much for watching, and I'll see you next time.